My name's Dale Thomas, and today it is a pleasure to be at Frontiers 2021 and pitching Mytide Therapeutics and discussing how we are combining manufacturing with machine learning in order to overcome molecular scarcity. Molecular scarcity is one of the main causes of the productivity crisis currently facing the pharmaceutical industry. And productivity has dropped 50% every decade since the 1950s, thus resulting in an astronomical cost of bringing a therapeutic to market. In order to overcome this productivity challenge, Mytide Therapeutics is focused on peptides as the therapeutic modality. Peptides sit in between small molecules as well as monoclonal antibodies. Peptides, however, have increased safety over small molecules and the selectivity of monoclonal antibodies, thus allowing them to treat a multitude of different diseases. The reason that peptides haven't been able to be accessed before is because of their slow and often long manufacturing times, even at the discovery stage. And in order to overcome this, Mytide has developed our BioFAB platform. And this allows us to produce peptides 30 times faster and at 10 times lower cost, thus allowing us to access data at scale and peptides at scales that nobody has ever been able to access before our platform. In order to accomplish this, we've developed a platform that really spans two different aspects. That is a hardware biofabrication platform, but as well as the software and data infrastructure that sits above the platform in order to capture information about every process step and every biological outcome in order to centralize that information, allowing us to use it to predict the characteristics of every molecule that we choose to produce. And we make these molecules 30 times faster than any other platform out there. The overall platform has been built, de-risked, and is in use 24/7 at our facility in Boston, Seaport District. The platform is broken down into three different components: upstream fabrication, downstream purification, as well as molecular processing. Throughout all of these different processes, we're able to produce around 1,500 molecules per week, and we leverage robotics at every step of the way. This is a platform that has not been realized at any other company or at this scale ever before. Mytide's platform is purpose-built in order to really enable both internal and external partnerships. Our platform has the ability to go after what we see as four main areas. Two of those areas are tissue-specific targeting and cell therapies, as well as protein degradation and monotherapies. We can go after this diversity of peptide therapeutics because each one of these projects is linked together through the data that we create for each one of these projects. We have established a growing external partnerships pipeline within cell therapies and drug discovery, as well as starting our own internal drug discovery programs. In order to do this, we've assembled a world-class team of scientists, engineers, and business development professionals, mainly hailing from MIT as well as industry. And we've established a scientific advisory board of well-respected professors as well, who are helping us accomplish our goal. We have launched a $10 million Series A, and this is really focused on allowing us to grow our platform and expand our internal pipelines. Today was great talking to everybody on this call, and really appreciate the mentorship that we've gotten throughout EFL. And we're really pushing biochemistry to its fundamental limits. Thank you.